Welcome to my course on microbial biotechnology. We are in module 2, which deals with the structure and life cycle of representative groups of viruses, prokaryotic and eukaryotic microorganisms. In this lecture number 3, we will be discussing about the structure of cyanobacteria and archaea. So, this is broadly divided into two sections, one dealing with cyanobacteria and the other dealing with archaea. So, we will start with the morphology of cyanobacteria, then also discuss in a little about the endosymbiotic theory and the great oxidation event. And in under archaea, we will be discussing about the ultrastructure of archaea, which includes the archaeal cell envelope and the archaeal cell membrane. So, let us begin with a discussion with the cyanobacteria. Cyanobacteria or blue-green algae are gram-negative prokaryotic microorganisms that lack a nucleus or membrane-bound organelles. They perform oxygenic photosynthesis, deriving their blue-green color from pigments like carotenoids and chlorophyll. Cyanobacteria come in three forms: unicellular, filamentous without heterocysts, and filamentous with heterocysts, the latter aiding in nitrogen fixation. They can also grow heterotropically, store phosphorus and nitrogen and form colonies with various structures. Cyanobacteria likely originated in freshwater or terrestrial environments and can produce gas vacuoles for buoyancy in water. Cyanobacteria display a broad spectrum of morphological variations, as you can see in these various pictures from A, B, C, D to H. So, in figure A, you can see the unicellular Syncocystis and B Syncococcus elongatus. In the non-heterocytous, you can see figure number C, which is Arthospira maxima, and number D Trichodesmium, and number E Formidium. And then you have the false or non-branching heterocytous. For example, in F you have nostoc, and in G you have Bracillonema octogenarum. And then you have two branching heterocytous, which are number H Stigmonema. Let us look into the morphology of cyanobacteria. Cyanobacteria cells vary in size from under 1 micrometer in case of Picocyanobacteria to up to 100 micrometer in tropical Oscillatoria species. Filamentous form shows cell differentiation with heterocysts for nitrogen fixation, echinates as dormant cells, and hormogonia as reproductive mobile filaments. These specialized cells and their connections mark the early signs of multicellularity in cyanobacteria. In this figure, you can see the formation of heterocysts, cysts, and the hormogonia and echinates in Anabena. Thylakoids. In cyanobacteria, the light-dependent reactions of photosynthesis occurs in thylakoids, which are membrane-bound structures parallel to plasma membrane with embedded photosynthetic pigments. Unlike chloroplast, these thylakoids lack grana stacking. Glyobacter bialysis is the only known oxygenic organism without internal thylakoids, housing its photosynthetic complexes in plasma membrane green patches. In this figure, we can see a membrane running parallel to the cell membranes as seen in Cenococcus, and in B we can see the converging membrane as seen in Synocystis species. So, these are two different variations of the thylakoid structure in a cyanobacteria. Next comes the phycobilisome. Phycobilisomes are protein complexes attached to thylakoid membranes in cyanobacteria, composed of an allophycocyanin core surrounded by phycocyanin rods, sometimes including phycoerythrins or phycoerythrocyanin. They act as light harvesting antennae for photosystem 2, absorbing light around 500 to 650 nanometer and transferring it to chlorophyll A. This allows cyanobacteria to utilize wavelengths that chlorophyll alone cannot, especially do in deeper water. So, you have this yellow phycocyanin and then we have here the chlorophyll, then we have the phycoerythrin in these rod sepate structures, and then attached to it are the phycocyanin. And then you can see light of various wavelengths getting absorbed by these photo harvesting system. Heterocyst or heterocytes. Heterocysts are specialized nitrogen fixing cells in filamentous cyanobacteria like Nostoc punctiforme, and then Cylindospermum stagnelli and Anabaena sphaerica. These heterocysts form at regular intervals, typically one every 9 to 15 cells. So, you can see here the heterocysts getting formed. Once differentiated, heterocyst cannot revert to vegetative cells. So, this differentiation is unidirectional. These cells convert atmospheric nitrogen into usable forms for other filament cells via the enzyme nitrogenase. Their role in nitrogen and carbon exchange reflects early multicellular coordination. Let us discuss about the adaptations in heterocyst. Nitrogenase enzymes are sensitive to oxygen. So, heterocysts have adaptations to protect them against oxygen injury. These include 3 extra cell walls, one being a glycolipid layer to limit oxygen entry. Photosystem 2 is degraded to prevent oxygen production. So, you stop the source from within. So, heterocysts rely on neighboring cells for carbohydrates, primarily sucrose. Heterocysts retain photosystem 1 for ATP production via cyclic photophosphorylation and use intracellular channels to retrieve sucrose. Glycolytic enzymes are upregulated to break down sucrose, and oxygen scavenging proteins are accumulated. Overall, all these mechanisms provide adaptations in the heterocysts for protecting the nitrogenase enzyme, which is sensitive to oxygen. So, here you can see the nitrogenase enzyme, and you can see here the transportation of sugar from neighboring cells. Heterocyst differentiation is triggered in nitrogen depleted conditions by the transcriptional regulator NTCA, which signals protein involved in the process. The differentiation sequence includes cell enlargement, reduction of granular inclusions, reorientation of photosynthetic lamellae, and development of a triple layered cell wall with the middle layer being uniform and inner layer laminated. So, this picture shows the division of labor in heterocyst cystus cyanobacteria. Echinid. Echinids are specialized dormant cells produced by certain cyanobacteria to survive harsh conditions like nutrient scarcity, extreme temperatures, and desiccation. You can see here the echinid of Dolichospermum and the echinid of Gliotrichia in the bottom picture. Larger and more resilient than vegetative cells, these echinids store nutrient and energy, enabling long term survival and dispersal until conditions improve. What are the adaptations in echinid? They have various mechanisms of adaptations. The first being development of a thick cell wall. Echinids possess a thick, robust cell wall made of complex polysaccharides and proteins, providing protection against desiccation, the loss of water, mechanical stress, and chemical damage. Another mechanism is that of dormancy. The echinids will enter a dormant state with reduced metabolic activity. and they have nutrient reserves. They store nutrients like glycogen and lipids, serving as sustenance during the dormancy, and they have pigmentation, contain carotenoids that protect against harmful UV radiation and oxidative stress from high light intensity. And they have germination control. Echinids can regulate their germination based on environmental cues such as nutrient availability, temperature and light, triggering the transition to a new vegetative cell. Let us now discuss about the Hormogonium. Hormogonia are mobile chains of cells produced by certain cyanobacteria during reproduction, playing a key role in dispersal and colonization of new environments. Here we can see nostoc growing on Z8 agar medium exhibiting vegetative filaments and Hormogonia under 40x magnification. These chains can glide across surfaces reaching lengths of several hundred micrometers and traveling at speed of up to 11 micrometer per second. By facilitating dispersal, Hormogonia help cyanobacteria colonize new habitats and expand their populations. How Hormogonia is formed? So, here is a schematic representation of Hormogonia induction and repression in cyanobacterial symbiosis. So, here you can see the filament with a heterocyst. Then due to Hormogonia inducing factors, there will be the formation of motile Hormogonia, and then we have symbiosis reconstitution, which will give rise to new filaments. The cyanobacteria differentiate into Hormogonia in response to environmental stress or when introduced to new growth media. This formation is essential for establishing nitrogen fixing symbiotic association with plants. When exposed to a Hormogonium inducing factor released by a plant host, cyanobacterial symbionts differentiate into Hormogonia. After approximately 96 hours, they revert to their vegetative state, ideally having reached the plant host to complete the symbiotic relationship. The endosymbiotic theory or symbiogenesis. Endosymbiotic theory proposes that eukaryotic cells originated from prokaryotic organisms. It suggests that mitochondria, plastid and other organelles evolved from once independent prokaryotes engulfed by host cells through endosymbiosis. Plastids likely arose when a free-living cyanobacterium was engulfed with the host, providing protection and nutrients, while the prokaryote contributed photosynthetic capability, so which was a win-win situation for both the systems. This relationship eventually led to the formation of chloroplast and the emergence of modern eukaryotic plant cells. So, here in this figure you can see endosymbiosis with two mergers creating eukaryotes and then plants. So, here the prokaryotes having archaea and then bacteria, the first being anaerobic and the second being aerobic. The first merger you can see of the anaerobic protists. So, here the mitochondria is being mobilized into the eukaryotic system, and this mitochondria is part of the animals and also as well as the fungi. Then this comes from the proteobacteria, as you can see over here. Then you have the cyanobacteria, which was photosynthetic, which got merged into the plant cells, and the plant cells also has the mitochondria. So, this is in brief the endosymbiotic theory or symbiogenesis, and this is how the eukaryotes probably acquired these independently existing microbes, and during evolution this becomes organelles inside them. Now, what are the proof of these kind of theories or endosymbiosis? Mitochondria and plastids can only replicate through binary fission. If a cell loses its mitochondria or chloroplast, it cannot regenerate them. So, these are two independent systems within the cell or eukaryotic cells, but the cells cannot produce them as such. Transport proteins called porins are present in the outer membrane of mitochondria and plastids, similar to those in bacterial membranes. So, which gives some indication that they were derived from these prokaryotic organisms. They are basically the prokaryotic organisms which have developed a symbiotic relationship with these eukaryotic cells. Some mitochondria and plastids contain single circular DNA molecules that resemble bacterial DNA in size and structure. Genome comparisons show a close relationship between plastids and cyanobacteria. Additionally, mitochondria and plastids have ribosomes that are more similar to bacterial ribosomes than to those of eukaryotes. Proteins synthesized by these organelles use N-formyl methionine as the initiating amino acid, similar to bacterial proteins, but different from those produced by eukaryotic nuclear genes or archaea. The great oxidation event. The great oxidation event marks a significant period in Earth's history, about 2.4 billion years ago, characterized by a substantial increase in atmospheric oxygen. Early photosynthetic organisms like cyanobacteria developed the ability to perform oxygenic photosynthesis, producing molecular oxygen as a byproduct. As these organisms proliferated, oxygen accumulated in the atmosphere, saturating oceans and lands, and triggering extensive chemical reactions and environmental changes. This rise in oxygen altered the atmosphere's composition, leading to the formation of an ozone layer that protected the plant's surface from harmful ultraviolet radiation. Additionally, oxygen facilitated the evolution of aerobic organisms, enabling more efficient energy metabolism through cellular respiration. Let us now discuss about the archaea. Archaea initially was mistaken for bacteria due to their similar size and shape, and were recognized as a separate domain in Wu's three-domain classification alongside bacteria and eukarya. They have genes and metabolic pathways which are more closely related to eukaryotes. Unique biochemistry of archaea includes either ether lipids in their cell membranes and diverse energy sources such as organic compounds, ammonia, metal ions, and hydrogen gas. As unicellular prokaryotes with circular genomes lacking spliceosomal introns, archaea exhibit co-located genes. Their DNA replication, transcription, and translation mechanisms are more similar to those of eukaryotes than bacteria. Distinct features include membranes made of isoprenoid chains, a motility structure called the archaeolum, and specialized metabolism like methanogenesis. Let us look into the structure of the archaeal cell envelope. So, unlike bacteria, archaea do not have a uniformly distributed cell wall. However, different groups of archaea share various cell wall components that are structurally similar to those found in bacteria. The physical properties of these components enable many archaeal species to thrive in extreme environment. In this figure, we can see different types of archaeal cell envelope structures from A to F. So, in some places we can see in A, for example, we have the S layer which is also present in B, C, D, and then we have in certain cases, for example, a protein sheet in B. Then we have this plasma membrane present all throughout. Then in C we have this methanochondroitin, and in F we have the polysaccharides. And in certain cases, is in the case of E, we can see outer membranes. So, this shows that the cell envelope structures of archaea are very, very diverse. Now, let us try to discuss some of these in little bit more detail. The archaeal S layer. Almost all archaea have an S layer, a protein based structure that serves as the primary component of their cell wall. This S layer consists of a single protein species known as S layer glycoprotein, typically ranging from 40 to 200 kilodalton and composed of multiple domains. In some archaeal species, the S layer is reinforced by polysaccharide layers, additional S layers or other surface glycoproteins. Depending on the species, S layers vary in thickness from 5 to 25 nanometers and contain uniform pores measuring 2 to 8 nanometer in diameter. In this figure, we can see the schematic illustration of the supramolecular architecture of the major classes of prokaryotic cell envelopes containing the surface S layers. Pseudomurine. Pseudomurine is named for its similarity to bacterial peptidoglycan. It consists of linear chains of alternating amino sugars and acetyl glucosamine and NAT. These are linked by beta 1,3 glycosidic bonds. Each NAT is connected to a short chain of L amino acids, typically glutamic acid, alanine or lysine, and these peptide chains are cross-linked to enhance the cell wall's strength. Pseudomurine is found in the cell wall of organisms within the methanobacterials and methanopyrales order. Methanochondroitin. Methanochondroitin is composed of proteoglycans with repeating trimeric units that include one glucuronic acid and two acetine galactosamine molecules. These GalNAc units are linked by beta 1,4 glycosidic bonds, while gluconoric acid connects to GalNAc through a beta 1,3 glycosidic bond. These are found exclusively in aggregated cells of methanosarcina. Methanochondroitin resembles eukaryotic chondroitin surfaces, which is reflected in its name. Other types of cell wall, some archaea like Ignicoccus, SM1 eukaryon and Arman possesses an additional lipid membrane outside the cellulosic membrane. Methanospirillum species have an outer proteinaceous sheet over the S layer with protein subunits arranged in a paracrystalline structure resembling stacked hoops. This layer can be separated under reducing conditions. Other species may have various polysaccharides including methanochondroitin and pseudomurine along with glutaminyl glycan in the halophilic genus Nephronococcus. Then we have lipoglycan in thermoacidophilic thermoplasma tails like Ferroplasma acidophilium and Thermoplasma acidophilium. Then we have heteropolysaccharides in Halococcus morhudae composed of sulfated units of various sugars including glucosamine, galactosamine, glucosaminuronic acid, glucose, galactose and mannose. The archaeal cell membrane. Archaeal membranes have a unique composition that distinguishes them from all other life forms, indicating a distinct relationship between archaea, bacteria and eukaryotes. So, here we can see a glycoprotein and some carbohydrate chains over here. Then there is a peripheral protein and transmembrane protein. All these are embedded in the lipid bilayer on either side. Like all organisms, the primary structural element in archaeal cell membranes is a lipid bilayer, as I have already told right now, which is made of phospholipids. These phospholipids consist of a polar phosphate head and a non-polar greasy lipid tail. Archaeal membranes are made of glycerol ether lipids, while bacteria and eukaryotes primarily use glycerol ester lipids. Ether lipids utilize ether bonds, whereas ester lipids use ester bonds. The stereochemistry of the archaeal glycerol moiety is the mirror image of death in other organisms. Archaeal phospholipids are built on a S-N-glycerol 1-phosphate backbone, which is the enantiomer of S-N-glycerol 3-phosphate found in bacteria and eukaryotes. This indicates that archaea use distinct enzymes for phospholipid synthesis compared to bacteria and eukaryotes. So, in this figure we can see the archaeal phospholipid, one the phosphate group, and then 2 the L-glycerol moiety, then number 3 the ether linkages, and number 4 the isoprene chains, and then in the bacterial or eukaryotic phospholipid in the bottom figure we can see number 5 the phosphate group, the D-glycerol moiety number 6, and the ester linkage in number 7, and the fatty acid change number 8. Let us look into the structure of the archaeal lipid tails. Archaeal lipid tails have a distinctive composition featuring long isoprenoid chains with multiple side branches and occasionally cyclopropane or cyclohexane rings. These branched chains help enhance membrane integrity by reducing leakage at elevated temperature. So, this figure you can see the archaeal lipid tails showing branched and cyclic isoprenoid chains in figure A, and you can see the lipid bilayer in figure B, and the lipid monolayer in figure C. Some archaea replace the conventional lipid layer with a monolayer structure, fusing the tails of 2 phospholipids into a single molecule with 2 polar heads known as Bola amphiphile. This fusion may increase membrane rigidity, allowing them to withstand harsh environments. So, with this we come to end of this lecture, which was focused on archaea and cyanobacteria. Thank you.